Hi, this is Roger Green, executive producer and host of the Surfing the MASH Tsunami podcast. This weekend, we are bringing you 6 conversations from episode 6, our Friday conversation with global KOLs about the impact of the Rezdiffra approval on Thursday. As the conversation starts, the various panelists describe their moods throughout the day on March 14th and their reactions when word of the approval came from FDA. They all describe feeling various degrees of trepidation as the day progressed, with Zobair Younossi probably feeling less so than anybody else, and some of the Europeans more so. This was replaced by excitement and joy at the final announcement. Panelists go on to describe the patients they expect to prescribe for Rezdiffra at launch, now for the U.S. when it becomes available in the EMA. Jörn Schattenberg centers in on F3 patients, but realizes he might not know exactly how to distinguish those from early-stage cirrhosis. Zobair Younossi points to using the F2 and F3 label. The approval of a first drug indicated for MASH may be the most significant achievement in fatty liver disease in longer than anyone can remember, certainly since some of the major diagnostic decisions and assessments were made. The conversation is interesting, and our global leaders have a variety of perspectives, so sit back, listen, enjoy, learn, and when you're done, join the conversation in our LinkedIn discussion group. Obviously, the most exciting thing that's happened this week for those of us in this part of the world is finally getting a drug over the line for the treatment of MASH, right? I remember at one point, the first time that obeticholic acid went down, somebody ran a headline in one of the business publications that said MASH 9 pharmaceutical 0 or MASH 11 pharmaceutical 0. I forget the number, but I remember the concept. Can't say that anymore. We haven't been shut out. In fact, we're off the schneid, right? So I just want to start by asking each of you where you were when you saw that there was an official approval and what your initial reaction was, what you thought about first.
Zobair Younossi.
I think most of us had been anticipating this and would have been short of a disaster if it wouldn't have been approved or would have been really very, very disappointing. So we were all pretty sure it's going to happen. The question was, exactly when the PDUFA date was the approval date was yesterday. So I've been looking for it all day yesterday and sort of interact with my colleagues with text, some of whom were really close to this process. So it was amazing when it happened, just because, as you said, Roger, this has been something that we've been burned so many times in the past, and that's been really a difficult challenge to me. And for that reason, and that reason only, not only that we have now a drug for a large proportion of patients that don't have an option right now, but also re-energizing this field of MASLD, or used to be called MAFLD and NASH, which was really getting a little bit labeled as a sort of impossible field. And I think to show that a drug can produce a great response was probably a very, very positive, not only for the efficacy that it has, but also for moving the field forward.
Points well taken. Next.
Louise Campbell.
I wasn't sure what time it was going to happen. I thought it would come out first thing in the morning, and it got delivered quite late UK time, so even later German time. And it was a case of let's listen to the webcast and listening to Stephen Harrison there. It took me back to the reason you guys developed the podcast was to keep, with COVID, people aware of these clinical trials, keep the recruitment going. This is testament to the fact that, did that work? Yes. Did we keep it in the agenda? Yes. So it's congratulations to Stephen and you and the team that originally started this, but also keeping people in the knowledge of this disease and the development as we were going along of fighting for any sort of knowledge base in the community. So that's where my thinking went first. Plus, it's not just a good day for fatty liver disease, but liver disease in general. As we start to look for one condition, we'll find fibrosis in other patients.
Duly noted. Oh, Jörn, you go next.
Jörn Schattenberg.
No, I was going to say quickly and then pass it on to Ian that was actually up the day in Amsterdam. I was a little nervous about the day. I've reflected on the journey, and Zobair Younossi is the better one to talk about it. I remember him presenting EASL data, Ocaliva talking about the watershed moment, and I thought, this must be happening again today. As he said, I expected it. The surrogate endpoints had been hit. This was the agreed conditions for conditional approval with the FDA. But on that day, I felt like, what happens to the field, to the patients, to us, if this is not approved? That nervousity carried me a little bit during the day. And I saw the AASLD message. I wondered about that too. So it was kind of a funny episode. I hope it didn't do any harm. And then after a full day, I think I was close to hitting close to my bedtime when actually I think 10:30 or 9:30 at night it came out. So I really just had a look this morning and studied the label even today. I know we're going to talk about that later. So I kind of slept through some of the excitement, and then I'm happy for us to have reached that point finally. I think the podcast went parallel with most of the clinical trial too. So it's really a great achievement, and I'm very happy to be here to be discussing results and the day after an approved drug in MASH.
Absolutely. Ian, go ahead.
Ian Rowe.
Yeah, I was at a radiology masterclass meeting yesterday with a room full of radiologists, and I told them confidently that this was going to be the most exciting day in hepatology for 10 years. There were a lot of blank faces looking back at me until I explained why that was actually going to be the case. It is really exciting, and even for those of us in Europe who obviously don't have access to treatment now, even within my sort of network of colleagues and friends, we were exchanging messages and real excitement about what was to come and what we were going to be able to learn from marketing in the US and the way that treatment will develop.
Jeff McIntyre.
We were at GLI, we were doing what a lot of other people in the field were doing, which was constantly refreshing our screens on most of the day. Having had this experience with multiple federal agencies, I can't say that we didn't have some concern the longer in the day it went, but we do have seen almost like their standard operating procedure is that they're going to make the big announcement late in the day. And so that it came before 4:59:30 that afternoon, I think was still earlier than what they're used to with this. And then we kind of spent the rest of the day, leading up to that, already beginning to field patient inquiries that were coming in, people asking about questions that are answered in the labeling, but, it will be part of our next steps and part of the engagement and conversations that we'll be doing, building on the live episode we just did with Louise on GLI Live to kind of start addressing some of these questions around access, about what the patient population is, et cetera, et cetera. Just the buzz was creating buzz on it, if you will.
Yeah, buzz tends to amplify. It's true, particularly good buzz. Jörn, I had the same experience you had at one point in the morning, thinking back to Zobair presenting Ocaliva data years ago and how excited we all were about that the first time and saying, no, that can't be happening again. So I spent the rest of the day in a state of what I'm going to call modified optimism. And the first thought I had when I saw the release was, phew, now we don't have to worry about this. We have lots of new things to worry about, but they're all good worries. In the battle of good worries and bad worries, we've now gone from the bad worries to the good worries. And the call I fielded right after that was from an investor buddy who was really impressed that we don't need to do biopsy, but immediately asked me, does that mean payers are going to require MRI PDFF on everybody? And I said, hey, take a deep breath. This is going to be easier than that. You got to step back from everything you were afraid of and approach it a little bit differently. And then, so here we are today. Hey, all good. So one of the questions I had for the treating physicians is when you actually read the labeling, actually everybody except me, when you actually read the labeling, did a specific patient or type of patient come to mind immediately as somebody you would want to have this for quickly, U.S., or as quickly as you can elsewhere, or something about how the target changed because of what was and wasn't in the labeling?
The obvious thought I had driving this morning is I want to use it in my advanced patients first, the ones that are F3, and I want to prevent cirrhosis. And then I sat back, said, I discussed with Ian a lot of times, how can I separate the cirrhotic patient using NITs from the non-cirrhotic patients? And then I had to take a deep breath and think about it and say, well, I need to put some more brains into that and decide on which NITs to be used. And I'm not sure, Ian, how you wanted to approach it, but I think that's the art moving forward, how to qualify the F2, F3 non-cirrhotic patients with the NITs we have.
Yeah, if I may add to that, I think that I thought about two patients. I thought about that patient that Jörn is talking about the F3 trying to prevent them from becoming cirrhotic. The labeling on this is very explicit about how it's supposed to be used in conjunction with physical activity and nutrition, or diet and exercise, as they call it, in that way as well. And so that made me think about people that are now moving into a state, F2, that are going to be more serious, but that it really pushes us into, okay, now we have a tool to help support the physical activity and nutrition efforts that you're making as well.
Well, a couple of things. First of all, the F2s and F3s are sort of the ones that I would certainly focus on. I actually think that we have to be very cautious not to go off label and include patients with more advanced liver disease. I think until we have better safety data, we should not really get ourselves into a situation where we have to explain things. And as you know, in the past, it has happened. And this disease, not this disease, but other diseases that you are familiar with drugs that are approved. So I caution people that we need to stick with the data that we have. And I think in terms of, I'll say what I would use for NITs. And I think this is really, as long as we follow the guidance that we have, how to determine the F2s and F3s using with the initial test. And then my second line test is that I really use the FibroScan or transelastography and ELF test as frequently as I can. And sometimes, we may have to use both if you have discrepant. I rarely go to liver biopsy, and I would not go to liver biopsy, and I would rarely go to MRE. That's because it's not really practical to do MREs in a lot of places because people are waiting for months to get MRI of their head for this or that. So it's not even actually capacity-wise.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingmash.com. We'll be back Wednesday night with a group of patient advocates discussing their excitement, reaction, and next steps after the approval of Rezdiffra. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.